Glycolysis is the first step in cellular respiration. It takes place in the cytoplasm of a cell, which is an aqueous solution that surrounds its organelles. First, glycolysis takes a glucose molecule. An ATP molecule comes and gives it an electron as well as a phosphate group. Note that to oxidize is to lose an electron, and to reduce is to gain an electron. ATP loses its phosphate group in a process known as ATP hydrolysis and gives it to glucose. ATP is also oxidized and glucose is reduced, means it gains an electron. This makes ATP, ADP, and glucose 6-phosphate. ATP has now only 2 phosphate groups, and glucose has 6 carbons and a phosphate group. Next, glucose 6-phosphate reacts with an enzyme. This enzyme rearranges glucose to make it fructose 6 with a phosphate group. So the glucose is rearranged, but it still kept its 6 carbons, and it still kept a phosphate group. Again, another ATP molecule comes in, and the ATP molecule is oxidized, and again, the fructose molecule is then reduced again. So it gains another electron, and the ATP loses its phosphate group. So fructose 6-phosphate becomes fructose 6 biphosphate, because now fructose still has 6 carbons, but it has 2 phosphate groups. Let's simplify the structure. It has 2 phosphate groups and 6 carbons. This can be split into G3P and DHAP. From here to here, a whole bunch of different enzymes come in and allow this split to happen. So now we have these 2 molecules. They both do similar things. They are both oxidized 3 times. We'll focus on G3P for now, but note that the exact same thing is happening to DHAP. G3P is oxidized, meaning it loses an electron, and NAD plus comes in and takes that electron. An electron from here is added to here, and whenever this is added, NAD plus becomes NADH. So since G3P has produced this by losing its electron, DHAP has also done it, so now we have 2 NADHs. Now that G3P and DHAP have both been oxidized, an ADP molecule will come in, and it will now be reduced, meaning now it's going to gain a phosphate group from this molecule, and it will become ATP again. Note that the process of ADP going into ATP is an example of substrate-level phosphorylation, where ATP is formed from the transfer of phosphate group from another molecule. In this case, it's G3P to ADP to make ATP. This happens twice. So since we have DHAP and G3P, there's been 4 total ATP made. After these series of oxidation and reduction, these 2 molecules form pyruvate, while also releasing 2 molecules of water. So from 1 molecule of glucose, glycolysis has made 2 pyruvic acids as well as 2 water molecules, 4 ATP, and 2 NADHs. Remember that we used 2 ATP at the very beginning, so we have a net production of only 2 ATP. Now that we have pyruvate, we can go into aerobic respiration, respiration with oxygen, or anaerobic respiration, respiration without oxygen. For now, we're going to go into aerobic respiration, and the next step will be pyruvate oxidation.
From glycolysis, we are able to take 1 glucose molecule and turn it into 2 pyruvate. To keep things simple, we'll use this structure. Now that we have 2 pyruvate molecules, we need to be able to turn them into the reactants of the citric acid cycle, which is acetyl-CoA. This is where pyruvate oxidation comes in. It takes the products of glycolysis and turns it into the reactants of the citric acid cycle. Here's the whole process simply. First, we need to transport the pyruvate molecules into the mitochondria. Remember, glycolysis takes place in the cytoplasm. We're going to get rid of 2 oxygen molecules and a carbon molecule, which will be carbon dioxide. Once you get rid of the carbon dioxide, you'll be left with acetyl. After that, NAD plus comes in and takes an electron from acetyl and turns into NADH. Now, acetyl has been oxidized, and the CoA enzyme comes in and turns the molecule into acetyl-CoA. Acetyl with the CoA enzyme. Now that we have acetyl-CoA, we can go into the citric acid cycle.
All right, I'm going to be explaining the Krebs cycle. Right now, we are located in the matrix of the mitochondria, where an enzyme called acetyl coenzyme A has been created through the process of pyruvate oxidation. Acetyl coenzyme A is a 2-carbon molecule that will combine with the 4-carbon molecule oxaloacetate to create a 6-carbon molecule called citrate. And this is done with the help of an enzyme called citrate synthase. Here we have our 6-carbon molecule citrate. And citrate is going to be isomerized into another molecule called isocitrate, which also has 6 carbons. This is done with the help of an enzyme called aconitase. Next step, we have our isocitrate, and it will be oxidized into a molecule called alpha-ketoglucarate. Now, if I could find, here it is, my alpha-ketoglucarate, which has 5 carbons. And this molecule, this reaction is helped with the enzyme called isocitrate dehydrogenase. You may notice here that alpha-ketoglucarate is missing a carbon. So what happens is this carbon molecule pretty much just is lost during the reaction. And it joins up with 2 oxygens to create a CO2 that is released as a byproduct that we breathe out. Also, during this reaction, 1 molecule of NAD will be reduced, gaining an electron and turning into NADH, which will be important later during the electron transport chain. Now here we have our 5-carbon alpha-ketoglucarate, which will be combined with an enzyme called alpha-ketoglucarate dehydrogenase into a molecule called succinyl coenzyme A. And you're going to notice here that again, there's a carbon missing, and again, it's for the same reason. This carbon is going to become a CO2, and it's going to be released as a byproduct. So far we have 2 CO2s that have been released. And again, 1 molecule of NAD is going to become a molecule of NADH. Through reduction, it gains an electron. So now we have our succinyl coenzyme A, which is going to change into succinate. And it's another 4-carbon molecule, but this time the enzyme is called succinyl coenzyme A synthase. Also, during this reaction, 1 molecule of GTP is created. Now, next here, we're going to have our 4-carbon succinate, and our succinate is going to be converted into a molecule called fumarate, another 4-carbon molecule, with the help of an enzyme called, one second here, the enzyme is called succinate dehydrogenase. And FAD will be created here, which will later gain 2 electrons and become FADH2. Now we have our 4-carbon fumarate here, which will then be created, be converted into malate, another 4-carbon molecule. So we're not losing carbons anymore. And this is helped with an enzyme called fumarase. Next, we have our malate here, which will, guess what? It's going to come all the way back around to oxaloacetate, another 4-carbon molecule. This time the enzyme, it will be called malate dehydrogenase. And in this step, 1 more molecule of NAD will be reduced into NADH. Now, if you think about it, we started with an acetyl coenzyme A and an oxaloacetate, right? Now, the acetyl-CoA coenzyme A is continually produced through the pyruvate oxidation process. And you'll notice that the oxaloacetate kind of goes around in a cycle, and it's, endlessly being created. So there's always kind of a storage of oxaloacetate within the matrix of the mitochondria. And that is the Krebs cycle. We have 3 NADHs being created. We have 2 CO2s that are going to be left. And we have one molecule of FAD that is converted to FADH2. I'm going to be explaining a process called the electron transport chain. So the first thing that we're going to need to know is that the mitochondria, here's my poorly drawn mitochondria, has two membranes. You can see them there. There's one squiggly one and one straight one. And the squiggly inside one is called the intermitochondrial membrane, and the outside one is called the outer mitochondrial membrane. And you can also see there's two spaces. In between the two membranes, there's a space called the intermembrane space. And in the very, very inside, in the middle, is another space called the matrix. So with that background information, we can get started. Here's kind of a blown up image of the mitochondria. Here's our two membranes. This would be the matrix. Here would be the intermembrane space, and here's the outer mitochondrial membrane. And so what's going to happen is two molecules from our Krebs cycle, FADH and NADH, both contain electrons. And these electrons will be ripped from these molecules, right? So now we have kind of just a free electron. And there's four complexes that lie on the inner mitochondrial membrane. Here they are. Let me just place them here, and they're labeled with Roman numerals from left to right. And the electron is going to move through these complexes into these very kind of complex patterns. Here goes our electron, it moved through, and that creates energy. It's the movement of an electron, right? Let's get these out of the way. And this energy is going to move a hydrogen ion from the matrix, here's the hydrogen ion in the matrix, to the intermembrane space. And when enough electrons move through the complexes and enough hydrogens are moved, we're going to have a gradient. We're going to have more hydrogens here. You could also say that these are just protons. They have a positive charge. We have more protons here in the intermembrane space than we have the matrix. As we know, it's going to create a gradient. It's going to create a charge. It's going to create a negative charge in the matrix, and it's going to create a positive charge in the intermembrane space. But the thing is that when you form this charge, something called, give me 1 second, I'm trying to remember. Something called an electrochemical gradient is created, which does not let these hydrogen ions pass back through the inner mitochondrial membrane. They can't get through. So what you need to get through, because they want to get through, right? Because there's more hydrogens here than are here. And so that's just how diffusion works. You need a protein called ATP synthase. And this is a transport protein that's going to take our hydrogens and it's going to move them from the intermembrane space all the way down into the matrix, right? And you might be thinking, like, this seems useless. We're just moving hydrogens back and forth, right? But through this process, ATP synthase has stored in it a molecule called ADP, adenine diphosphate. And when this hydrogen moves through, it creates energy, which lets the protein transport ADP and make it ATP, which is how we store energy. It creates 32 ATP in this process. It's an ATP making machine. And this movement of the hydrogen is creating ATP for our cells to use as energy. One more thing to note here is that when the hydrogen moves through the semipermeable membrane, it's called chemiosmosis. Think of osmosis as usually when water moves through a membrane. Chemiosmosis would be when a proton moves through a semipermeable membrane. So that is the electron transport chain. It produces 32 ATP.
Fermentation occurs in anaerobic conditions or when no oxygen is present. There is no electron transport chain involved in fermentation, and there is no Krebs cycle involved in fermentation since oxygen is not present. Since oxygen is not available as the last electron acceptor, we need a new electron acceptor, which is where fermentation is involved. So in glycolysis, glucose yields 2-pyruvate, and in this process, 2-NAD is reduced into 2-NADH. It's also important to note that 2-ATP is produced in the process of glycolysis. Now, NADH, the electron carrier, would normally be delivering the electrons gained to the electron transport chain if this was aerobic cellular respiration. Once losing their electrons, NADH would then be oxidized into NAD+. But there is no electron transport chain involved in fermentation. So now we need to find a way for NAD to be regenerated for glycolysis to continue again without oxygen or electron transport chain. This is where pyruvate becomes the electron acceptor. Pyruvate accepts the electrons from NADH, which then yields lactate. In this process, the 2-NADH is then oxidized into 2-NAD. NAD is regenerated now, so glycolysis is able to occur all over again. This recycling of the 2-NAD and 2-NADH is able to happen because the pyruvate acts as the electron acceptor. This happens in lactic acid fermentation, which happens in our muscle cells when we're working out and oxygen cannot be delivered fast enough. So then lactic acid builds up, which is when soreness occurs. But in alcoholic fermentation, which our bodies cannot perform, acetaldehyde, a derivative of pyruvate, is the new electron acceptor. CO2 is released in glycolysis in alcoholic fermentation. Lactate is not produced, but ethanol is. But NAD is still being regenerated and used again in the process of glycolysis. But this is alcoholic fermentation.
What happens whenever these processes are disrupted, though? It's very rare that these processes are disrupted. However, whenever they do happen, it can be pretty deadly. For example, methylglyoxal. It's a highly toxic compound, and its biggest characteristic is that it's an agonist to glucose, meaning it mimics it. Now, methylglyoxal is actually a waste of the human body. So whenever there's a big buildup, it can harm the body, causing lung, colon, stomach, and breast cancer, as well as just being really generally bad for your body. So whenever the buildup is not removed, it is very harmful. So what happens is that it builds up and it takes over glucose's spot in glycolysis. And whenever glycolysis isn't able to perform, then the Krebs cycle or citric acid cycle is not able to continue, and therefore the electron transport chain isn't either. So your body's just not respirating, and it's very, very bad.